All right, so today we will be presenting Dr. Heinrich. Dr. Heinrich, he is the man. He is a distinguished professor at OHSU, and he's a part of the Life Raft Group Advisory Board. He is renowned for his groundbreaking research and clinical expertise in oncology. He's been with the Life Raft Group since the beginning, and we truly thank him for all his work. And today he will be presenting on the new landscape for GIST, specifically clinical trials. All right, and Dr. Heinrich, you can take the mic.
Thank you. It's a pleasure to be here. I was noting as I was running this morning that it was much more pleasant to run in the dry air than it was running in New Orleans 2 years ago when the humidity was like almost made me pass out. It's a pleasure to be here. I've been, as noted, I've been working with the Life Raft Group, I think for like 24 years since I first met with the team in a Holiday Inn in Wayne, New Jersey, way back in the day. So we're kind of, I guess, doing this a little bit backwards because we just talked about what are the new advancements, what are the new clinical studies, and I'm going to tell you more what is the standard of care. I try to level this so that it could be just 101 for some people and just 601 for other people. So I try to provide background, but also some of the more higher level concepts for the gisters who have heard it all already. So these are my disclosures. And so the major themes that I'd like to go over would be the molecular classification of GIST, and then talk a little bit about how our drugs were developed, because they use different approaches which kind of would lead us to interpret the data differently. Most of the drug studies early on were mutation agnostic, which means you have GIST, you get the drug. And more recently, we're doing what we call precision oncology, which is you have a mutation, we will give you a certain drug. And that's led to different routes of how drugs are approved. We talked a little bit about persistence and hibernation in the last session, if you were here, so I'd like to come back to that. And then finally, I'll close with some open questions to which we would desperately like to know the answers. So, just by way of background, GIST is a type of sarcoma. It probably comes from the cell called the interstitial cell of Cajal, which are the pacemaker cells in the wall of your stomach and your intestines, and that's what makes food and digestion go from basically one end to the other and not back and forth. The most common site of a tumor is the stomach, but it can arise in the small bowel, the large bowel, the rectum, or various other less common sites. And we think, although we still don't know the answers to this, I think exactly, we think that there's about 4,000 to 6,000 cases that are medically significant. That means that you had surgery and we discovered them. And prior to the year 2000, we didn't have any treatment at all. But coming from those early days, 2000, we now realize that even though for a long time in the last 20 years, we've treated all GIST like they were the same, we now realize that that's not true. They're not all the same. It's true that they are the same tumor type and the pathology diagnosis is the same, and they all come from the same starting cell, but they differ in terms of what caused them. And most of them come from a mutant kinase, and I'll explain what that means. And the important point is that knowing the type of GIST that you have completely determines what therapies will work or not work. And so this year, we have kind of a robust classification of GIST. So in the pie chart, the big blue is the KIT mutant GIST, which gets most of the discussion and most of my discussion today, because that comprises almost 80% of GIST. But then there are a couple slices in orange and mauve, I don't know, some desert color that represent PDGFRA mutant and sdh-deficient GIST. And there's a parallel session going on right now, I think, about sdh-deficient GIST, but those are about 10% each. And then at the top is a bunch of really tiny slices of things that are very rare, 0.1%, 1.5%. And then, as we talked about last session, these days only 1% of GIST or less are unclassifiable. That means that we can't find a mutation no matter how hard we work. So since we talk a lot about mutations, I thought I would maybe go back to GIST 101 and talk a little bit like what is a mutation and what is our lingo? And so really we're talking about the fundamentals of molecular biology, which is that DNA gets transcribed to eventually make RNA and then make a protein, and that determines function. So you could think about that in terms of, well, it's sort of a language of the gene, and so how does that relate to language of other things? So a series of letters comprises a sentence, and when we read the sentence, we derive a meaning. What does that sentence mean? And so we can start with a string of DNA letters, and we could say, well, the meaning of this is that the cat is fit. So that DNA ultimately determines function because it determines what the protein is like. But if you change the DNA, you can change the protein, so you can change the function. So you could change one letter in our sentence, and the cat is no longer fit, the cat is fat. And that's an example of an abnormal function mutation. So in terms of understanding mutation reports, the way I think about it is as a library. So a library is comprised of all the books. That's all the knowledge. So that's your genome. And in that library, one specific book is analogous to a gene. So we have the book of KIT. And so that book is all about KIT. And in a book, we have chapters, which are units of organization. Well, in a gene, we call that exon. So when we say that there's something wrong with KIT exon 11, what we're really saying that is, of all the genes, we're only talking about KIT, and now we're only talking about chapter 11. And then the specific mutation that's mentioned in your report is analogous to a typographical error that's in your book that changes the meaning. Now, not all mutations completely change the meaning, so cat with a C versus cat with a K, it's not quite correct, but we would still understand what you were talking about. But if you changed it more profoundly, so cat could be one letter changed to fat, rat, car, or cab, that would completely change the meaning. And those are what we call a change of function, whereas other mutations like cat to cat are silent mutations. They're not normal. They're not the way it's supposed to be according to the book of DNA master code, but the meaning's not changed. And so then when we apply that to GIST, if we look at the two most common mutations, KIT and PDGFRA, comprising about 80% and maybe 10% respectively, then we see where these chapter things come in. So in the book of KIT, in GIST, we really are only talking about exon 9, 11, 13, and 17. And when we talk about PDGFRA, for the most part, when we're talking about primary mutations, we're only talking about 12, 14, or 18. So what do these mutations do? So these mutations that cause GIST change the function. So KIT is what we call a receptor, and so the kind of basketballs that are floating, two lines of them, that's the cell boundary. So the top, the golden part of the protein sticks out of the cell. And then down below is the business end of KIT, which is a kinase, which means it transfers phosphates to a protein, is located. So normally, KIT is fine. You need KIT. In fact, if you don't have KIT, you actually will have problems. So you want to have KIT. You just don't want your KIT protein to be active all the time. So here's an example of an inactive KIT. But sometimes, like in your bone marrow or in other parts of your body, like pigmented cells, we want KIT to become activated. And so we send out a hormone called KIT ligand or KIT LG, and that causes two of these molecules to come together. And when they come together, it kind of bends the protein, and then it becomes activated, and then it sends a signal. And so if your body needs KIT to be functioning, it sends out the hormone, and as long as the hormone's around, it will signal. But if the hormone goes away, KIT will not be working. So it's either off or it's on. And the problem with mutations in GIST is that it turns it on. So here in this example, the protein's on, it's signaling, it's telling the cell to not die, it's telling the cell to grow, but yet there's no hormone, it's inappropriate. And furthermore, it's on all the time. So it's kind of like the accelerator in your car that you can't take back. It's stuck to the floor all the time, the car is out of control. So these are the type of mutations that cause GIST. And so, when we think about beautiful places and basics of real estate, it's location, location, location. When you think about your GIST, you really should think mutation, mutation, mutation. So really, you really need to know your mutation. So example, my name is John Smith, and I have a KIT exon 9 mutation. And so I recommend mutation testing really for any situation in which your doctor is thinking about doing something other than surgery, thinking about giving you a drug. So if you had surgery and they're thinking about giving you a drug after surgery, you would need to know your mutation. If your tumor is unfortunately come back and spread, it's metastatic. And so before treatment decisions should be made, we really need to know your mutation information. And as it was discussed in the last session, increasingly clinical studies are quite restricted. To be in a study, you have to have a KIT mutation, otherwise you're not eligible for that study. So let's talk next about treatment of metastatic disease. So a little audience participation. So let's have all the gisters put their hands up. Okay, so put your hand down if you think there's only one approved drug. Put your hand down if you think there's only 2 approved drugs. Put your hand down if you think there's 4 or fewer. Put your hand down if you think there's 6 or fewer. Okay, everybody put their hand down. The answer is 10. There's actually 10 drugs. The reason that you don't know that is 5 of the drugs are approved for the KIT or PDGFRA mutant GIST, so that gets you up to 90%. But the other 5 drugs are approved for forms of GIST that are less than 1%. And we'll talk a little bit like how did that even happen that they could get approved. And so our journey to find therapies is kind of had used 2 different models. And so the studies that led to treatment of imatinib, sunitinib, regorafenib, and ripretinib, if you had GIST, you could be in those studies. And it wasn't restricted to mutation. And I would call that mutation agnostic. And that is kind of good and kind of bad design. It's good in that KIT mutations are quite common. So that means that probably 70 to 80% of the people should be on these drug studies. But it also means, and we'll talk about this, that 20% of people have mutations that could not possibly benefit from those drugs, but they were included in these studies. And so when we look at what the FDA puts out to tell doctors how to use drugs, it doesn't say that you have to have a KIT mutation to say you have a drug. It only talks about how many drugs have you already taken, and it's okay for you to switch to the new drug. And more recently, for these drugs that are much less less common forms of GIST, they actually were what I call precision oncology, in that you could not be in the study unless you had a specific mutation. And for the context of GIST, it didn't matter that your mutation is only 0.1% of all GIST patients. If you had that mutation, you could go in the study, and the drug eventually could be approved for your treatment. So, again, imatinib is, our original therapy, and it works quite well, and these probably even works better than what I'm showing here. I think I should also explain at this point what a graph is showing. So this is an example of survival outcomes. So this is an event-driven plot. So the y-axis is always telling us something. It's either overall survival, which means when do people pass away, or it's talking about progression-free survival, which is when does your tumor get worse. So at the beginning, at time 0, 100% of people are alive, but as this curve goes down, that means that somebody in this study passed away, and that makes the curve go down. And you can see that even after 14 or 15 years, there's still about 20% of people who are alive, which is rather remarkable from where we started, but not nearly good enough. So then we move to our second line drug, which is sunitinib. Now we've sort of changed it up. Now we're no longer talking about overall survival. Now the y-axis is how long until your tumor got worse. And we're no longer talking about years, now we're talking about weeks. So sunitinib in the second line works on average for about 6 months or 27 weeks. And then we rolled into regorafenib as a third-line therapy, although some people, it was a fourth-line therapy. And again, we're looking at progression-free survival, like how long till your tumor got worse. And again, we're talking 4.8 months compared with placebo. And then our most recently approved drug, ripretinib. But remember, because it's kind of important as a patient perspective, that in these studies, any GIST could have been in there. And if we knew then what we knew now, we would have designed the study, and you would have had to have a KIT mutation to be in the study, and then we would have a result that was more reflective of what we really want to know in making decisions. So, why are some GIST resistant to treatment? Well, sometimes it goes back to us. What is your mutation? So really, in all the studies I just ran through really quickly of the approved drugs, imatinib, sunitinib, regorafenib, and ripretinib, we're giving it to all the GIST patients, but really there's subpopulations that do really well, like a KIT mutant GIST, or certain types of PTGFRA mutant GIST, and then all the, fortunately, more rare forms of GIST, they would not respond. And they could go from first line to fourth line therapy without having any benefit, and that is historically how we designed the drugs. So what is happening when we treat, so that's primary resistance, like we give people a drug and it doesn't work from the get-go. Usually that's because they had the wrong mutation. So, but what happens when we treat people with something that could respond? So we give imatinib, which is the appropriate therapy to someone who has a KIT mutation. So in this, analogy, you have this beautiful life outside and you want to go through this door and get to the beautiful life. But when you get a KIT mutation, the door is closed. So we can't get to where we want to be because of the locked door. But fortunately, we have Gleevec or imatinib, which is really just a key to turn KIT off. And we can open the door and we can restore, our beautiful life and health. But as we treat for longer, and this was discussed in the first session this morning, you get more and more mutations, and they're harder and harder. Now, maybe you don't need one key, maybe you need 3 keys, or you need a third key that can open the first 2 locks. So, and it gets more and more difficult. And the reason, and this is kind of my version of resistance mutation, so we're kind of looking at the KIT protein on the left side, kind of ignore all that, and we're talking about the drugs on the right side, and we're kind of lining them up against different mutations. We talked before about exon 13 and 14 as being one class of resistance mutation, and there are 17 and 18. So if you look at, for example, imatinib, red means it won't work. It doesn't work, and these are the mutations that lead patients' tumor to become resistant to imatinib. The problem is when you switch to sunitinib, what happens next depends on what tumors you have and what mutations. So if you have tumors with the green mutations, sunitinib will work very well for you, probably much better than average, and it might even last for years. But if you have tumors with the red mutations, sunitinib probably won't even work for 2 months. And the problem is that we don't really know who has what in what tumor, and generally people have a mixture. So you have some sensitive mutations, you have some resistance mutations. So when we treat with sunitinib, we see, oh, those things are getting better, that's great. Those things are getting bigger, that's not great. And we're trying to decide, weighing the pros and cons of the net change, whether we think it's still helping you enough to continue. But if we switch to regorafenib, we sort of then, we change the script, but we don't change really ultimately what happens. Some mutations are resistant to regorafenib, some are sensitive, and when we switch to ripretinib, we see the same kind of thing. Certain mutations are resistant strongly, and other mutations are sensitive. So none of these drugs are really what we regard as optimal, which is the far right hypothetical situation that no matter what mutation is present, we would have a drug that works. And that's still what we're trying to get to. Just to kind of flesh out a couple of examples. So again, here's the idea of PDGFRA mutant GIST, which are in the two boxes in the green or the red. And historically, when we gave you therapies like imatinib, we sort of had a split outcome. Some people did great, and some people did totally not great. And you can sort of see this in this survival plot. Again, this is time to tumor progression from when you started therapy. So the dark line has a specific mutation called D842V. It's strongly resistant to imatinib. The curve goes almost, I guess I don't have a pointer. Oh, sorry. I've been pointing to things, but I guess it doesn't show up for you. Okay, but this works. So the curve goes straight down, which basically means that the first time your oncologist did a scan, they would conclude that the tumor had already grown, and that most people, almost everybody, by 6 months, their oncologist had decided that this therapy was no longer working. But other forms of PDGFRA mutations that weren't that actually did quite well. And some people, this is what, 8 years, and still like almost a quarter of the people are 8 years into the therapy and it's still working for it. And you know, again, this is sort of a failure of taking everybody and putting them into the same box when the box, really they should be in two boxes. And so when it came around to developing a better drug, avapritinib, they no longer said, we're giving this drug to everyone. The design of the study was, we're going to take people with GIST who have a D842V mutation, because we know it doesn't respond to the other drugs, and you have to have that mutation to be in the study. And these are the results. So this is, again, a waterfall plot. So each bar is an individual patient. We're tracking their change in tumor size from baseline, so you want it to go down because that's getting smaller. And you can see remarkably that in this study, some people had complete response. All of their metastatic disease was gone, could no longer be detected. And almost everybody had what we call a partial response, which is a 30% decrease in diameter. But if you did the math, because 30% doesn't seem super impressive, decreasing the diameter by 30% is like decreasing the volume by, like, I think 75%. So it's a profound change in size. Think about getting a 16-inch pizza versus a 10-inch pizza. Like 6 inches doesn't seem like that big a deal, right? But 10 inches like feeds you maybe, and 16 inches feeds a family of a certain size. And so these small differences, when you start, you know, squaring them or cubing them, add up to a big difference. Another example of, again, how the original design didn't get us to where we want, but new designs can, would be the case of NTRK fusion GIST. So NTRK fusion GIST are very rare. It's probably less than 0.5%, maybe it's 0.1%. There's actually a little bit of controversy about whether some of them are actually GIST or they're a different type of sarcoma. But let's assume, since this is just a presentation, that they're GIST. And so, for example, there's something called NTRK3, which is just the book of NTRK3, and it can be mutated and become activated. And so this is an example from Dr. Siklik of someone in an early phase study of a drug called larotrectinib, who actually had been on all of the approved and then some other standard GIST drugs and had not responded to any of them. And then when they got as a 6th-line therapy, larotrectinib, which targeted the specific mutation that they had, then you could see on a PET, so this, oh, that's right, you can't see that. This big thing is a tumor, and the bright spot is the radioactive tracer, which means this part is all metabolically active, and the center of it's kind of dead because it grew too fast. And after 8 weeks of therapy, really it's much smaller, and you don't see any metabolic activity at all. And so because these type of events or mutations were rare, we had to come up with a different clinical trial design. And so in this trial design, we're again looking at a waterfall plot. This is taking a whole bunch of people with NTRK3 mutations, and this is the list of diagnosis. So GIST are the red bars, and then there's lung cancer in here, colon cancer, breast cancer, other soft tissue sarcomas, pancreas, all kinds of different things. And so rather than a trial of GIST patients, this is a trial of patients with NTRK fusions, and they're treated with the drug. And in this study, there were only 4 patients with GIST. But because the study was overall positive, the FDA said, we are approving this drug, and you can use it for any tumor that has this type of mutation, including GIST. So, historically, if we were doing a study of imatinib and we took 4 patients and we tried to go to the FDA and say, hey, we treated 4 patients, give us a drug, they'd say, well, now go treat several hundred and give us some more data. But in these new designs in precision oncology, we lump people together on mutations, and if the study is overall positive, you get approval. The ultimate sort of outcome of this was the approval of agents for BRAF mutant GIST, of which I think there's exactly 4 reported cases ever with drug treatment. And in the study that led to FDA approval, there was only 1 GIST patient, and they were not even a valuable response. But nonetheless, thanks to the design, we now have these drugs approved for GIST if we need them, even though historically we'd say a study of 1 patient doesn't prove anything. So this leads to the major theme, which is the importance of knowing your mutation. So I've kind of flipped the paradigm. Before it was, here's the drugs, and we get outcomes below based on what your mutation is, sort of sorting things out. Here we flip the script and say you should know your mutation first and then get your therapy. So if you have KIT mutant GIST or certain forms of PDGFRA mutant GIST, then we should go through our standard imatinib, sunitinib, regorafenib, or apretinib. And if you have D842V, you should avapritinib. Here's the BRAF approval. And in fact, the repotrectinib was just approved, I think, 10 days ago, so it's the 10th GIST drug, again, by a similar kind of study. I don't think there were actually any GIST patients in there, or if they were buried in the nomenclature of sarcoma, and they haven't really explained it. So if you have the green box ones, then you should get the appropriate therapy, and we would expect that the drugs have a high likelihood of helping you. If you're over here in sdh-deficient or NF1 or FGFR mutant GIST, we still kind of don't know exactly what to do, and probably clinical studies that kind of go after these mutations is probably the appropriate thought. And my final kind of theme of the day is the difference between persistence and resistance, or hibernation versus resistance. So this is a study kind of back from the early days of imatinib, when we knew that it worked for GIST, but we didn't know as much about it. And we were coming from a world in which treatment was more chemotherapy-based, and so we can't give chemotherapy to somebody for 20 years. Like, we might not be able to give it to you for more than 3 or 6 years, or months. And so the thought was, well, all these people are doing really quite well at 1 year, 3 years, 5 years, or 10 years of imatinib therapy. Do they really need to continue? Because we don't typically continue therapy for a lot of cancers indefinitely. And so this was a randomized study, And so the patients in red were those that after 3 years of therapy, their disease was controlled. So we weeded out all the people who didn't respond to imatinib. These are the responders. And they continued therapy. And so there is some fall off of the curve, which means that some people are having progression. But that curve in red is a lot better than the blue curve. The blue curve is the people who stopped their therapy. And even though they'd had great scans for 3 years, after they stopped therapy, generally within a year, or certainly mostly by 2 years, almost all of their tumors had started to regrow. And this curve is really teaching us two different things. So the upper curve is resistance, so secondary resistance. The drug is working, the tumor mutates and finds a way around it. But the bottom curve is really telling us about persistence. It's proving to us that no matter that your tumors were controlled for 3 years, they're still hibernating cells and they don't have any problem, for the most part, waking up. So disease persistence is those cells that remain viable during drug treatment and they can resume growing when you stop the therapy. And so certainly in the metastatic setting, those hibernating cells are the cells that if they mutate 10 years down the road, that's where we get resistance. But this also applies to some of the discussions we had the last hour, which is after surgery and you had a high-risk tumor, and we give you therapy, but after we stop, it comes back. It also applies to the setting where we don't even know that you have tumors, but if we stop your therapy, your disease can show up. But resistance is different. Resistance is when your tumor grows despite the fact that you continue on the drug. And then this is sort of the kind of life cycle of treatment of GIST. So somebody with metastatic GIST, we give them imatinib, so the number of imatinib sensitive gets smaller and it keeps going. But eventually, unfortunately, over time, you get these resistance mutations, and so then we have to switch to other therapy. But persistence is all the stuff that happens over here. So it's before you start therapy, while you're on therapy, the disease can keep hibernating and we never get rid of it. And resistance is when your tumors get new mutations and start growing despite therapy. Now, it turns out that we can model this in the laboratory. So we can take GIST cells, as shown here, treat them with imatinib, and count how many cells are left. And over a 7-day course, you can get rid of 95% of the cells. But if you stop the therapy, then the curve goes back up because all those cells that survive will keep growing. And it turns out that we've done this experiment for a week, we've done it for months, some people I think have done it for a year, that no matter how long you treat the cells with imatinib, always some cells survive, and as soon as you stop therapy, they will grow back. So theoretically, we could use that to model it. This is kind of a complicated slide. The easiest part to interpret is looking at the spots. Spots are the tumor cells that we're staining, and if you do this experiment where you treat them with a drug and stain them, you always have some cells left with all our favorite drugs. But if you do combination therapies where you use a KIT inhibitor plus different inhibitors like a MEK inhibitor or a PI3K inhibitor, you can theoretically in vitro get rid of all the cells. And that's what we kind of need to aspire to. I'm not saying that this is what we should do. This would probably be extraordinarily toxic. But we could at least shows theoretically it's possible. So kind of here's the summary, 10 drugs have been approved, but we really need to know what your mutation is to be able to use them. And then I could leave this up for just kind of thought of these are the questions that we talked a little bit in earlier sessions and we'll talk about in later sessions of things that we don't know about but we wish you did. Thank you.